The overall goal of the following experiment is to identify proteins specifically interacting with a given target protein in cell extracts containing soluble and membrane proteins. First, achieve full metabolic labeling of chlamydomonas cells with nitrogen-14 and nitrogen-15 and generate cell lysates containing and lacking ATP. An optional cross-linking step helps to stabilize transient protein-protein interactions. Next, separate the cell lysates into fractions enriched in soluble and membrane proteins, from which the target protein, its specific interaction partners, a control protein, and contaminating proteins are immunoprecipitated and eluted. Now mix the immunoprecipitated proteins and triptychally digest and analyze them by tandem LC-MS. The results distinguish between true and false interaction partners by showing different nitrogen-14, nitrogen-15 peptide ratios for proteins specifically interacting with the target protein, and equal nitrogen-14, nitrogen-15 peptide ratios for the target protein, the control protein, and contaminating proteins.
The method of choice for the analyzing protein-protein interactions is to immunoprecipitate the protein of choice directly with its interaction partners from cell extracts, where both occur at their native conformation and concentrations. However, when doing so, also contaminating proteins are precipitated mainly because they interact with the antibodies used. As modern mass spectrometers are extremely sensitive,